Até já. Tu vais ouvir daqui a pouco. Tu daqui a pouco vais ouvir.
No, this one. Hello? Yep. No.
Ok. Vamos então dar início a mais um seminário do mestrado em Informática Médica. Por uma questão de cortesia, vou mudar para inglês. Welcome to a new seminar of the Master in Medical Informatics. Today is our pleasure to have here Dr. Dietrich Rebholz-Schuhmann from the University of Zurich. Dr. Dietrich is a well-known researcher and has a research group devoted to computational linguistics and some knowledge discovery from biomedical data. And I'm pretty sure you will enjoy this seminar. He will be with us until Monday. We will have an informal research discussion at the Faculty of Medicine next Monday. So any of you that would like to attend, it's free to come. Please do. We will have, I hope that this will be a nice add-on to this seminar. But for now on, I would just like to thank you for being here and to giving this lecture that I'm pretty sure everybody will enjoy. Thank you.
Yes, many thanks, Pedro and Altamira, for inviting me to present my work. So the first time I get broadcasted, so I'm a little bit nervous in this. So I speak English. I speak not very fast. I hope you understand. If you have questions, just ask. I will present you several things. One thing is, what is a phenotype? The second thing is how we use semantic resources, terminologies for text mining, literature analysis. The third thing is looking into the future, how we use them, how we generate new information. So for my background, I'm a computer scientist and a medical doctor, but I'm working mostly on the medical informatics, bioinformatics side. And for my past work, I've worked on an institute of bioinformatics. So I'm coming from the bioinformatics corner. And I've done a lot of literature analysis. I've built search engines. And these search engines are still available on the web, so it's easy to find. But I don't talk so much about text mining here. I talk about how we use these resources to integrate data, to find new knowledge. And I focus in particular on phenotypes because this is what is at the moment the most pressing and the most interesting. So all the projects where I'm working on, we try to find new knowledge by defining what is a phenotype. So I will explain what a phenotype is, but a phenotype is really difficult. So a phenotype can be very complex, but I give you an idea how we deal with them right now. Again, if you have questions, just ask them. So I give you a little bit of motivation, then I explain what a phenotype is, and then I go into the analysis, how we use them, and then comes the nicest chapter, what do we do in the future with this? What is going on right now, what we have to consider in the medical domain when using semantic resources. When I talk about semantic resources, it's all computer-based. It's something like a terminology. It can be more complex than a terminology. It can be a vocabulary. So these are all things you can write down by hand. You can collect the terms. You can put them into a file, and you can use them afterwards. And I reuse whatever I get from all over the world, from everybody. I'm working together with the people who do this. I exploit everything. This goes direction of semantic web, but it can be as simple as using terms in a text. I give you an example, so you will see this, how it works. So first, I define what is a phenotype. And I said already, this can be very complex. So phenotypes is something like, so this is something how we describe an organism. If you look at a person, you can describe the skin, you can describe the hair color, you can look inside of the organism. But there's something what we call model organisms. So these are certain mice, rats, certain fish, and they are analyzed systematically. So these model organisms are observed. So you take a mouse and then you put it into an experiment. It runs over a bridge and then you observe whether it's more anxious or not, whether it falls down or not. And then you write it down, and this is put into a database. So we describe certain model organisms, but we also want to describe medical patients. So this has happened over the last 2,000 years. By observing a patient, we know whether he is okay or not, and we write down what is not working, functioning. And in translational medicine, this is currently going on. We want to judge a patient, but then we want to use research from molecular biology to use this knowledge to find new drugs. And that's why we want to know exactly which genes are inducing which phenotype. And then we do data integration. So I use information from molecular biology and want to use information from the patient in the clinical domain, bring this data together, and then find out whether a gene doesn't work for a certain patient. I can look up the genes for the patient, but I want to know whether a phenotype is influenced by a certain gene. So what is a phenotype? I said already it's very complex. So a phenotype is something which we can observe, a trait. And so by observing the person, we can write down what the phenotype is, how it looks like. hair color, eye color. And certainly a phenotype is induced by the genotype and the environment. So genotype is, or the genes, and you know that some diseases are induced by the genes. For example, sickle cell anemia. The gene produces a different hemoglobin, and then the red blood cells look differently, but also by the environment. And then you look for clues to understand the functioning of the phenotype. So we look into the genes and want to find out whether they induce a phenotype, and we look into the treatments that a person receives. And if we can put everything together, then in principle we can predict the phenotypes. So this is for the future, not for now, but the genotype plays a role and the treatment plays a role. And so they may look quite different. So if you, for example, take a human or a mouse, they have a different phenotype. So a mouse doesn't look like a human and a human doesn't look like a mouse. But the genetic background is very similar. Mouse and human have 96% similar genes. And funny enough, they don't look alike. And why this is the case, difficult to explain, but we have to resolve this. So ideally, we could take a mouse, do an experiment with a mouse, and predict what a human how a human will react. This would be the ideal solution. And I give you an example of how we bring these resources together. So I have an example which is quite difficult, but it shows the problem. So we have aging. Aging is also a phenotype. Every person ages. So it's the accumulation of changes in a person over time. So we change over time, or phenotype changes, and this is what aging makes up. But how do we define it? Well, it's not a disease. Probably. We all age, but we are not sick if we age. We normally age. But the changes induced by diseases make aging. So is it a process? Well, aging looks different for all the different people. And so all the changes that happen in a body probably induce the aging, but it's probably not a single process. The changes are neither positive or negative. So it's something we all age, but it can be positive to age, it can be negative, we don't know this. So is it a phenotype? Good question. Somehow we can describe a person as being old or young, so aging must be something like a phenotype, but it's very difficult to define. So we can say that everything that belongs to aging is a phenotype, but aging as such is probably the sum of everything that makes aging. So this is a complicated example. It's easier to say somebody has blue eyes or somebody has green eyes, but these are typical problems that we are facing. We want to define how a person changes over time. Okay, so this a little bit provocative example, aging is a phenotype, but it's very difficult to define. Now I make it a little bit easier. So what is a phenotype? And we have defined, if you look very carefully, we find things which look kind of abstract, but are very straightforward. So anything that is a physical object can be a phenotype. So a liver is a phenotype, and a spleen is a phenotype, and a brain is a phenotype, and a bone is a phenotype. And then if you have modifications to the organs, then this is also a phenotype. So if an organ is bigger or smaller, so an increased liver size is a phenotype, and a smaller liver size is a phenotype, or missing red blood cells is a phenotype. Then function is a phenotype. So whether or not the blood carries oxygen is a phenotype. So people who have the normal oxygen saturation in blood is a phenotype, or whether they have problems to pick up the oxygen. And then there's biological processes. So this is something like metabolism. If we can digest glucose, or whether we can digest proteins, or whether we can build certain substances. So these are the phenotypes. So this is an abstract definition, but everything in the body, in principle, can be defined like this. So this is a cool thing. And I will explain you later how we use it. So here's a couple of examples. Increased and decreased size of an organ, cells, tissues. So this is a qualitative phenotype like megalosplenia. Then the increase or decrease of functional, physiological, and metabolic processes. So if the heart rate goes up or down, whether the blood pressure goes up or down, whether the blood glucose tolerance goes up or down. So this is what a medical doctor would do. He would sit down, ask the patient, you have high blood pressure, and if he has a high blood pressure, then this is the phenotype. And not everybody has a high blood pressure, so only some people have a high blood pressure. Then existing or missing structural components. For example, if there's a connection between vessels in the lung or in the brain, this is something which is abnormal, we can describe this, and then molecular abnormalities. So you see already from these examples that there can be quite a lot of different phenotypes, but this is ongoing. A medical doctor writes them down and then uses the description to identify the patient. So, okay, my aging example again. So we have reduced physiological functions. So the tissue perfusion goes down, tissue regeneration goes down, lower O2 perfusion in the brain. So this is what we find in an older person. We have reduced metabolic functions, pre-diabetic conditions, increased blood sugar due to reduced insulin activity, and we have changes to the body structure. So lost elements, hair, teeth, kidney. Hair is a good example. So losing a hair is normal. Everybody loses hair. But, okay, you can have it also at a younger age. So it's not even, you see, it's difficult, but older people have less hair, but it's not a characteristic of old people only to have less hair. Okay, so maybe I should change these examples. I should probably switch to teeth. Teeth are probably a little bit safer. Okay, so how does these phenotypes look when we want to work on them? So I'm interested in electronic resources. So what I defined now is my theory, and I would like to use the theory on real resources. And now I show you the real resources. They look different. They are not as clean as what we think in our theory. So what I work on my theory is something I use to write grant proposals and to use a certain model. But this is reality. So we find text where the phenotype is in. So here we have the Angelman syndrome, and then it contains a description, and the Angelman syndrome is a neurodevelopmental disorder. I have to understand what this is, and then there's a couple of other parameters which are here. So the phenotype is in the text. And I know how to deal with this. I can read this as a human, but I can use also computers to read them. I can make them, analyze them automatically. This is cool, but I won't talk much about this here. I give you an idea how it works, but I could go on for hours on this. This is my core domain. But what we have is that the same text is what a medical doctor writes down. So he describes the phenotype, and then we have to take it and we have to analyze it. This is probably what you know. This is in the electronic health records. And the language used there is quite complicated. It can be very cryptic and very difficult to read. But in principle, a medical doctor writes down the phenotype, not standardized in free text. The next thing, how we find phenotypes, is this. And you may have heard this, SNOMED CT. SNOMED CT is a terminology. And this is an organization which collects descriptional phenotypes. They have a lot of people, they write them down in different languages, and they make them available. It's a tree structure, it's an electronic structure, we can use them. We can use the terms in there, but we can use also the structure. And what's quite nice, they are also in different languages. So we have used them for a project where we wanted to translate from English versions into French or German. So we use them to standardize multilingual resources. So this is already a little bit nicer to use. It's nothing where I have to use text mining. I just download it, I use it, and apply it. And there's something which is a little bit more complicated, and you may not have seen this. So this is a graph, and this is there are several terms in there, and these terms are describing also the same thing as before, phenotypes, but the structure here is highly elaborate. This is something which we call ontology or a taxonomy. So people use their understanding of how phenotypes are structured to make a really excellent logical structure into this. And so this is very useful. So you can say things like the brain is located in the skull, and the brain is used for thinking. And you can use this, you can describe in a very detailed way how the phenotypes look like. The difference to this structure is that here we can use a lot of reasoning. So we can find out whether an organ is in the located close to the stomach, and so we can use this to reproduce the knowledge that a normal person is doing. So again, all these are phenotype representations, free text. You find this in the literature, in the patient records. Then there's a lot of collections of terms which we can use. And then there's something which we call an ontology, very complex and developing and very useful. And here's another, here's a comparison between all of them. So here we have the text, here I have the vocabularies, and here we have the ontology. And from here to there, slowly. So this part is for humans. Humans can read them easily. And this part is more from computers because they can analyze them automatically. And I show you how we use them. So phenotypes is something I explained in the beginning, can be quite complex, can have different features, and we find different representations for the phenotypes. Here's again my picture, which I've shown you, the physical objects, liver, spleen, brain, then the qualitative genes, increased size of a liver, increased size of a heart, functioning, whether blood carries oxygen or not, and biological processes like metabolic functions. And what I've added here is the resources which we use. So for all of these elements or these definitions that we have, we find electronic resources which we can use. So we grab them, researchers are developing them, we exploit them, we download them, we use them. So physical object is chemical entities, cellular components like a nucleus of a cell or a cell membrane. And then there's anatomy descriptions, liver, bone, everything. Then we have qualitative themes, so human phenotype ontologies. So there's several resources, but there's also still some gaps. So not everything is available, but so we use them, and I give you an idea how we use them. But if you look inside of them, then we would love to see that something which is described on a mouse is the same as for a human. You've seen 96% of the genes are the same, but unfortunately a mouse has a fur, so it doesn't work quite like this. But if you compare them, you find interesting things. So for example, in the mouse, this is human phenotype, this is mouse phenotype, and then we have hearing loss in both cases. Wonderful. It's just the same. So hearing loss in the mouse is the same as the hearing loss in the human. Hopefully it's the same. I'm not sure how they ask the mouse whether she's hearing or not, but they have ways to do this. And but then we find things like progressive childhood hearing loss. This is for humans. And in a mouse we have abnormal hearing physiology or abnormal ear physiology or hearing vestibular ear phenotype. So they're all different. It doesn't really fit. It's not talking about childhood. It's something which is abnormal. It's not a loss. So the question is whether the loss is the same as abnormal. So a loss is certainly abnormal, but not everything abnormal is a loss. So there's kind of a mismatch which we have to resolve. And then you have something like an optic nerve hypoplasia in human. And oh, this is abnormal. Abnormal optic nerve morphology. And this seems to be quite different. So we have done a systematic comparison, and although these resources are really cool to use for us, it's not yet the case that they all fit together. So I have to do a little bit of text mining to get them together. And so how do we exploit them now? So we have all these resources. I told you that we want to do experiments on model organisms. We want to do experiments with mice, and then you want to see how it works for humans. And so we use them by doing comparisons between human and mouse. So we can analyze what is known for a mouse and then use this for a human. We can find out whether a certain SNP is a singular nucleotide polymorphism, is a single modification of a gene. And we can use information about the SNPs from databases and then find out whether a certain drug is having side effects. We can analyze complex phenotypes, and altogether, if we take these resources, we could read what a patient has in terms of problems. So in principle, we can look up from the phenotype what genetic background is involved. But I give you examples how we use them in reality. So how do we normalize? Now I give you a little bit of a text mining story, only to show you how it does work. It's not a medical text, so it's something which is easy to read. So the patient records is something which we can analyze and read, and a lot of terminologies are available. So in principle, we take a patient record and then look up the terms and do some text mining and statistical methods, and then make this available. And I have to admit, I haven't worked on patient records that much. I've worked mainly on scientific literature, but I'm currently preparing projects where we apply the same thing for patient records. And but others have done this. It's only that I wasn't in the position to use it on patient records, but the methods are the same. So analyze the patient records, and one thing we have done this not on patient records, but on the scientific literature, and I will show you how it works. We use the terminology, and then we standardize the patient record or the literature. So we look up all the terms, and we use the identifiers. And if you use a semantic web approach, we use URIs. So we normalize the patient record. We pull out everything that we find there, make a profile vector, and then use this. And here's an example. So this is how it works. So it's not about a patient, it's not about disease, it's about sumo and sumo wrestling. And it explains all the examples which we have to deal with. It comes from the scientific literature. So this is a typical example I would find. So you find sumo is a competitive full contact sport where a wrestler, so this is the whole text. It's about sumo wrestling. And the typical thing is you look up the terms. Sumo is something which is very specific, something which we wouldn't use every day. And then we find other terms like rikishi, which is also very special. It seems to be the wrestler here. We find sumo bout, which is the ring. We find something like wrestler. So this text seems to be about wrestler. And soles, feet play a role, competitive full contact sport. And then we have all the terms which are specific. And then we do a little bit of statistics. And then we have something what we call a profile. So this text is about a wrestler, force, ground, feet, first, opponent, ring, sumo, touch. It could be even medical. So feet is medical, and touch is medical, and force is something you would use in medicine. So we find this profile, we find these words, and then we can describe the text by these specific terms. And this is all pretty straightforward. There's only a couple of problems you have to consider. So this is also a little bit about text mining. So sumo is martial art. But sumo is also suggested upper merged ontology. So this is an ontology. Then it's also a small ubiquitin-like modifier. This is a protein. Then we have sumo is surgery and molecular oncology. This is a department of the Dundee University. And it's also software update monitor. Mozilla supports sufficiently uniform memory organization. So it's a lot of things. Spacecraft for the unmanned modification of orbits. So there's a little bit of a problem. When we use terminology, we have to make sure that we're talking about the right thing. And so although sumo seems to be very straightforward martial art, it's also a medical term. That's why I pull out this example, because this is what I find when I do medical analysis. So apart from using these resources, we have to be a little bit cautious to find the right terms, the right semantics. And there's some effort which goes in. And so this is what I've done in the past. I took all these terminologies, which you know, species, chemical entity, gene and proteins, and then I take an engine which analyzes the text, and then it finds something like a chemical entity. And this chemical entity, aspartyl tRNA, is part of a synthase, which is a protein, and then there's a gene, which is the gene name, and then there's a species around it, and I analyze it, and then I know what the person is talking about. So this is a more complex example than the sumo example. But again, use everything that we know from the public and apply it to the text. And this is how the engine looks like. It takes an object in the beginning, and then looks for the sentence, and then looks for the different entities, the terms, and then looks for the different concepts, and then makes a really big document. So if you look up the engines, the search engines, which are labeled with my name, they all work with the same principle. There's a pipeline, and this pipeline takes all these terminologies, finds them, and extracts them. And these terminologies is big, so we are talking about something like 20 million terms, something like this, and so far more than any human person would ever use and know. And so a normal person who's an academic knows maybe 50,000 terms, but we are talking about something between 2 and 20 million. Okay, so this is what we do with the terms, and so we use the terms and we categorize them. Here's an example for proteins. So there we have looked into how protein names can be interpreted, but I don't go into detail here. This is another problem that we are facing when we apply the terminologies. So we have mice and mice protein. So somebody founded a clever idea to call a protein mice protein, which is actually not a clever idea because the mice protein is not saying anything. So it's not a very well chosen name. The other problem that we're facing is we find something like left breast cancer. And then if you take our terminological resources, we find breast cancer and left breast. Now the question is whether a breast cancer in the left breast is different from a breast cancer in the right breast. And this is even for a medical doctor difficult to say. There may be a difference, but at the moment I wouldn't say there is one. And then we have something like retinoblastoma, which is a disease and a gene. And so difficult to distinguish. And then we have something like Streptococcus pneumoniae, which is a species. And we have Streptococcus pneumonia, which is a disease. So the difference is one letter here at the end that makes a big difference. So I know that you won't develop terminologies, but these are the big things that we find. So now I show you how we use this. So initially I said, what is a phenotype? Then I told you we have the different resources, and then we apply them to text. And now I show you an example how we use this. And this is an example I've done together for pharma companies. So this is a real project that we have done. So we used genes for diabetes, and then we looked up what functions are influenced by the genes. So we looked up the Go terms tell us which functions are influenced. And we also looked into phenotype mentions, which are linked to the genes. And then we looked for those functions which are linked to the disease and to the gene. And we looked both up in the literature. So we find gene which is influencing a function, and the function is broken in a disease. And then we find the link between the gene and the disease. And this is what we have done. Again, big numbers. So we looked up disease names, and we built a triple store here. So this was the CESAR project. It was about diabetes mellitus. And every number here is a single statement from the literature, whether a disease is linked to a gene, or a gene is linked to a function. And we generated from the literature, about 20 million statements, and these statements were reporting about relations between a gene and a disease. And this is what we found. So we take genes from one database, which are linked to diabetes. So this is a reference database, which reports on genes for diabetes. And we look into another database where we find genes which are linked to pancreas by gene expression. We look into the literature. This is where we find that a gene is linked to a disease by different means. And then we look into another database, which is reporting about all the genes in the proteins and telling us whether a protein is linked to the disease. And then every number here is a single gene linked to any of the two diabetes. And then we can look in the overlap, and we find interesting things. So these are all the known genes for diabetes. This is not interesting. This is also known knowledge. So this is all the proteins which are known to diabetes. Easy. So remains this one here, this number here, and yes, this number here. So these are all genes which are known to be expressed in diabetes, but they are not known in the reference data resources, and they are known in the literature. So this 19 is only known in experiments and only known in the literature. So this is knowledge discovery. So you pull them out, and you think that this is a clever thing. These are probably known genes, or new genes for the disease, and then you look them up one by one, and then you decide whether this is a good target. And these are the genes, so this is the same list with the names. Okay, so this is the one example I wanted to show you how we use the literature in combination with the other data resources and use the terminologies to find links between genes and diseases, and then afterwards we validate them against the reference data resources. Okay, so much for the experiments. Now I go a little bit into the future. So I show you what is going on in our heads, what we would like to do, what we would like to get out, what us and others would like to get out. So this was the example which I've shown you before. We take a disease and we take a gene, and then we look up all the links between a gene and a disease. So this is the database with experiments. This is the database which contains all the description of diseases. This is mouse experiments, and this is literature. And down here we have a gene and the mouse phenotype. We link the mouse phenotype with the human phenotype, and then we find the link between the gene and the disease. So this is what you've seen in the previous image. So these are different sources to link the genes to the diseases. And here we use the terminology for the phenotypes, and then we can compare what we get out of it. And and this is a little bit more in detail. So here we have the disease, and then we look up all the genes that are, all the phenotypes that are linked to the disease. We find all the phenotypes which are linked to the genes, and then we compare them, and then we get a certain result. And this is what we found. I don't go into the medical part here. I want to move on to the future. Do I have to 6:30, or when did we start? Okay, so this is also a little bit more complex example. So here we have one example which is bringing all the different resources together. So this is what I've shown you before. So we use the different phenotype representation, anatomy, and the quality, and the process and the function. And then we bring them together, and then use the information from mouse and from human. And so here are the ontologies, and here are the databases. So we take all the diseases, link them with all the phenotypes, and then we use a reasoner which defines us what phenotypes are linked to, what genes are linked to the diseases. So this is a very special result where we use actually the ontological structure. And here is another example where we have used the phenotypes. So here we have used something which is the, so here we are talking about drugs. And so we take a drug like tramadol or pantoprazole, and then find how these drugs are linked to a certain function. So we can look this up in a database. And this function here can be linked to a physiological function. So this is something which is human or mouse. So this is a special function in the body. So we had made a link between a chemical function and a human function, or a medical function. And then we can, because we have this link now between the chemical function and the physiological function, we can find out whether drugs have the same mechanism. So we can look up whether drugs which are known here and have this physiological function, and drugs which are known here and which have a chemical function, whether these are related to each other. So this is a method to find out whether a drug which is known to one disease could be used for another disease. So if we know that a drug has a certain human function, and then this human function is linked to a chemical function, and we know that another drug has this chemical function, then we can find out whether these two could be replaced. So for this one, this is a true positive where both is given, and these are, unfortunately, we also have false positive and false negatives. So this is the example I've shown you before, where we go over the different structures. This can be a little bit more complex, so we can look up certain, so we take the human phenotype, we can look into functions which are linked to the human phenotype, and look up the genes. So this is a little bit more complex example. But I just want to go on. So this is the example for the electronic health records, where we would like to use them and use laboratory data, and then link the disease types to the genetic level. Okay, so, but these are the scenarios for the future. So now this was work that we have done. Now I look into the future. So I'm using all these all these terminologies and technologies to map resources. And now I give you an idea where this would be leading to. So if we take these resources, we have all these terminologies, we can describe what the phenotype is, we can describe a patient, and now we want to use this in real life. And these are things which are partially happening, these are things which are ongoing, and where this would fit in. So here, we could improve diagnostics. So we could help a medical doctor to take a diagnostic decision. So how would it work? We take the patient records, analyze it, and then we look up, we compare the patient phenotype against the disease. So we say, this is what we found, and a disease is defined like this, so does it fit to the disease or not? Systems like this exist, but they don't use phenotype representation. So this would require that we have to define the parameters which make up the patient, but it may happen in the future that we use genetics too. So we can analyze the genes, we can do this at reasonable costs, which is maybe still a bit too high, but coming down. And so we can take what the description is from the medical doctor, we can take the laboratory results, we can take the genetic parameters, put everything together, and then suggest a diagnostic. And this has to be shown that this works, but this is called translational medicine. So this would be decision support for a medical doctor, but it could be also to help patients to read their own data. This is the next thing that can be done. So we can identify side effects of drugs. And this is similar to what I've shown you before. We take a drug, we find out what it influences positively and negatively, so whether it has a positive effect on metabolism, physiological parameters. So we make a profile of this drug, and then we look up whether, if a patient becomes 2 or 3 or 4 drugs, whether this positive effect or negative effect is generating side effects. And this is really a difficult thing. So a typical patient in Zurich at the hospital during a single stay gets 21 different drugs. Out of these 21 different drugs, you have to know anyways which ones interact, but you may have to know for a single patient whether this patient has a special problem. I've once, in my old days when I was working at a medical doctor, I was taking part in a what was it, a gastroscopy. And then a guy came up and he got his normal sleeping drugs, more or less, and he got the normal dosage and nothing happened. And then he got twice as much and nothing happened. And then he said, okay, I know this, it always happens with me, I'm not a drug addict, I just need more. And then he had to get more. And this is not an easy decision, it's something which is quite risky. And so in this case, finding out about side effects would be a good thing. And the point is that some people have a different metabolism. Some people have a slow metabolism, some have a fast metabolism, but it's difficult to figure out. And certainly all side effects between the drugs is another thing which we could figure out. And this is what would be an interesting thing to resolve. Then the next thing is what we call systems medicine and OMIM 2.0. So if we look into a patient, then we can describe a disease with the phenotype profile. And then we can use all the different resources and describe how a disease functions. And so we can define a model of the disease. And the disease is asthma or diabetes, and then a patient gets a certain drug. And then we can compare against the profiles of the drugs against the disease profile. So this is going even more into the future. Well, actually, I gave this talk about a year ago, but right now I think the future is catching up. So this is IBM Watson. I'm not sure who has heard about IBM Watson. Nobody? Yes, you did? So IBM Watson is a computer, and this computer is quite clever. So it uses a lot of facts. And it has a very good success in a show, which was a quiz. So the Jeopardy quiz. So this quiz is, you take all the facts that you know, and then you state a fact, and somebody has to ask the question where the fact fits to. So it's really complicated. You have to ask who in 1749 died in some kind of Viking ship in the North Sea by, I don't know, something like a meteorite which blew off the boat, something like this. And so they invited the 3 best Jeopardy winners into the show, and then IBM Watson was the counter player, and he won. So it shows quite potent technology. And now they try to get this into place for diagnostics, and this is what I've explained before. So using all the facts about diseases and about the genes, and then feeding a computer, and this computer has to resolve the diseases. And at the moment, it's using schoolbook knowledge, and they try to be as good as a medical student when he starts his studies, but they are not yet there. So still a little bit, I still have a little bit of hope that I can be faster than them. Then the other thing which happens is big data. Who has heard about 23andMe? Who knows 23andMe? Nobody? 23andMe is a company which is associated with Google. Actually, the wife of one of the founders of Google has founded 23andMe, and it's a company which offers sequencing. So at the moment, it costs $5,000 to go there, and then they analyze all your 23,000 genes, and then they keep all the data, but they tell you what's in the data. And so it costs $5,000 to get the complete analysis, $99 to get a small analysis, and so they keep a lot of data, they do the analysis, and then they send you a report whether you are sick on some kind of serious disease or not. So this is going on, and my judgment is they need 5% of the population to know the whole population, and I think they're probably already pretty close. So we will know all this data in the near future. And then the other thing is patients like me. Anybody heard about patients like me? No, no, no. So this is another company which is in Boston. Yeah, social network is an interesting point. So it's they want to be a social network, but it's more a medical advisor. So they ask people to submit their patient data. It's an American company, so certainly in Europe people wouldn't do this. They would keep their patient data for themselves. But in America, there's a company who advises people or patients. So they submit their patient data, and then they get advice on their disease. And certainly it's a little bit a critical thing, because who knows what they do with the data. Okay, so these are the things which are going on, but this is all going the same direction. So we will have genetic information, we will have disease information, it will be in the public to some degree, and depends on the national restrictions. And certainly the data integration that we are doing is adding to this. Okay, so my summary. So the semantic resources are the key element. So this is something which exists, we use it, and they help us to standardize information. It helps us to normalize the information, to use it. Large data is not the problem anymore. So nowadays, computing power is available. We can make use of large volumes. I still remember about 15 years ago when we started to work on Medline, it took us about 3 or 4 months to analyze it completely. Today, we do it in less than 2 or 3 hours. So, and the biggest problem is semantic integration. So what I said from the beginning, what is the phenotype? How do we define it? How do we use it? This is the tough thing. How do we shape our knowledge? How do we make it available? How do we make it consistent? And then this is the tough work. This takes time and time and time. But once we have this, using profiles, statistical methods, automatic reasoning, this is the interesting part. So all of a sudden, the data grows together. And certainly, semantic web helps for this. And then the key development is that patient records will be brought together with molecular biology and genetics resources, and to understand what is in the data. Yeah. So this is my talk, and I have time for questions.
My first question, well, first of all, I think it was a very, not only interesting, but especially inspiring talk, because we can, through your talk, have a glimpse, really, of the future. Phenotypes are not only complex, and but they are almost infinite things. You mentioned about 20 million, but with the progress of science, could actually become 200.
It could, yes.
because if it was 100 years ago, it would be much less, because of course the knowledge generates more knowledge, and we can subdivide and be more specific with the time it goes on. So the thing is that this kind of approach, as many other approaches, is probably a continuous type of thing, that we'll never reach the thing, but we can, on the way, doing much more useful things than we are. So, and especially enjoyed and liked the aid using these techniques to help diagnosis, and probably also to help, which is probably what pharma companies are more interested in, to help the personalized medicine, to be able to target more specifically the different phenotypes. Of course, you have, this will be further complicated by the polymorphisms and all this, but the thing is, when do you think that we'll have because I think the moment, there will be a moment where all this will be exploding.
I think, well, in some sense, we are limited. We have only a limited number of genes, but we have a larger number. Even the number, if you take all the polymorphisms, the number looks big, but there is only 8 billion people. So, okay, big number, but this is finite. So if you compare this against chemistry, chemistry is infinite. So there's an infinite number of chemical entities, but there's only a finite number of humans, and there's only a finite number of genes, and there's only a finite number of diseases. Well, I'm not quite sure. So I believe there's only a finite number of diseases. So getting this equation sorted out, this is doable. And I think my judgment would be that if you take the decision point in the clinical domain, this is not a huge thing. It's the decision point. So for example, my best example is always schizophrenia and depression. These are two different diseases. We are maybe not 100% sure about this, whether they're clearly different. But if you look at the treatment, it doesn't make a difference whether somebody is a little bit provocative, but the treatment is more concerned by the symptoms than by the exact diagnostics. And I think this will happen. I think I'm even more tough or more provocative on this statement. The pressure which comes only from the costs on the healthcare system means that we won't explode decision-making, we will reduce decision-making. And this is something where, at the moment, we don't have the handles. So it's very difficult to do this. And one thing where we have some hope is that the genetics may make a handle to reducing costs.
But just another thing, but on the other hand, well, there are already very good ways of systematically analyzing, and even cheaper and cheaper, the genetic part. But what about the description of the phenotype part?
No, I think we still have a big hurdle. So there's something like a hurdle which we have to go over. This is the mountain. We have to climb the mountain to find afterwards how easy it can be. So every new technology will do this. And I don't know how big this mountain is. But I think my judgment would be there's a couple of diseases which are where we always knew they are genetic. So arthritis, for example.
Yes.
There's something where you can feel it. You know this. There are different categories, but we don't know how to separate them. We try to do this by the symptoms. We try to count how many limbs are involved, whether jumping or not. And you have all these measures already how to capture this, but we don't know how to fix it. And now all of a sudden we have this means to fix it by the genetics. So the number one example currently is acute myeloid leukemia. So there are, I think it is 6 different types which have been identified by the genetic causes, and they make a difference in treatment, and this is what everybody's waiting for. It's only so difficult to get this. So you have to run the clinical trials, you have to get your cohorts, you have to do the mathematics, you have to understand which genes are involved. So this is the hurdle we have to go over, but we will find this. And whether the more nasty thing if I put forward this argument is that you have so many diseases which are induced by the patient themselves. So there's still a little bit of a training going to happen for patients.
But in any case, to be able already to model so many things, especially the physical, in the physical domain, because then you need to model also some behavioral things. Yeah. And but it's very promising. And,
well, I think you will always try to find the decision point. So you don't model everything. This will never happen. So you model those pieces where you can see the decision point. So if I take arthritis again, you don't take the most remote 2 types of arthritis. You take the ones which are closest and where you can see that you need some kind of decision point. And then you think about your theory and why this is involved, and then you model this part, but only this part, and then you take a decision here.
But the influence of environment, it can be so strong in some of the disease that in one way or another you need somehow to model this, because at some environments some disease will never have symptoms, and in other environments they will be pretty severe.
I think you will always try to ignore environment, and it may be a big mistake.
Yes, but I think without really putting environment into question.
No, I think what will happen is that you take drugs which have an influence whichever genetic background, because then you can have your experiment on the human. So you have the disease, the treatment is allowed, then you do your study, you find out that the metabolism plays a role, the genetics plays a role, and then you try to get your cohorts together so that the environment plays a small role. But I know this, there's diseases where it will be very difficult.
But maybe then you can somehow try to absorb the problem with the environment by studying let's say course in different countries at different social backgrounds and so on. This then will be working for some and not maybe really working for some others.
Yeah. But I think it can happen in a completely different direction, which is probably the social networking thing. So if you look around, then patients know themselves best what is influencing them. Mm-hmm. The problem is they have to go through this channel to the MD to find out what is now wrong. And it could happen if you have a more properly represented disease description, like with IBM Watson, people would explore this, and then they themselves would argue, oh, I have more this or more that. So pain is a very good example.
They would be a better doctor themselves.
So yeah, well, pain is a very good example. There's so many different kinds of pain, but if you're an MD and you ask somebody for pain, it's pain. It's difficult to find out. But if a patient describes, no, it's more like this, located here and there, they can find out amongst themselves what is the pain they're having. So that's my assumption.
Thank you.
Thank you. Any other question?
So would you say that in the future we don't need physicians?
Oh, we always will need physicians, but I think they become more and more expert. I think well, if you look at the work of a physician, he would like to get rid of everything that is the boring stuff where he doesn't need his expertise. He wants to do the interesting. When I go to a physician, I sit there for half an hour, we discuss all the laboratory results, because this physician loves to discuss with me what's wrong and what's right. So this is, I think, the normal things where you have to go through the normal things all the time. This is the boring stuff. So if you look in other countries, like in, well, I can only compare Germany and England, then in England you have different levels of expertise, and the expert only comes in the last stage. And there's a different way how to deal with this. And in Germany, it's more that they have to deal with all this day-to-day work. This is not a good thing. You would like to get rid of everything that is only unproblematic, and you only want to deal with the problematic cases. Right, Joshua?
Why would we need the physician if we have all the information, or if we can have almost of the information by ourselves?
Yeah, very good point. So why do we need a best friend? Because we talk to somebody. So whenever I have a software problem, I have to explain to somebody else to find out what my problem is.
Only if you don't have the solution, for instance. So
now the MD gives the expert feedback. So if you take a decision which drug you're supposed to take, then this is a very tough decision. So how to take an expert decision is something you cannot read from. You cannot even read it from a book. You need the expertise. So if you have a child and a child has hearing problems, you go to the physician. He knows he knows whether this is an infection which just starts off, or whether this is something which has a more chronic behavior. And there's a big difference. And he may know that this is currently around. So this viral or type of influenza is currently around. So the expert here has a lot more knowledge, which we won't be able to. We can do the straightforward things. But we cannot take the decisions. We cannot give by a computer the decision to a human. This wouldn't work. So the physician will always be more expert.
So there is always things that we cannot put in the computer.
I think we can cover
almost
a lot of knowledge, like the 80%, 90% thing, but the tough decisions we cannot do. So a very good example is if you have a pre-birth judgment on deviation on a child. So if you have a child and you do like the sonography, and then you have a 1 to 200 chance that there's a disability. Is 1 to 200 a lot, or is this little? So what do you do with 1 to 200? You have to take a decision. You have to take a decision whether you now do more invasive analyses, and then you have to know whether the risk of the invasive technology is 1 to 200, higher or lower. And then you have to take a decision whether you want to have more diagnostic. And this is very tough. So this is maybe the main problem. If you do what I've been suggesting, you get these long lists of probabilities. But humans cannot do. I can deal with probabilities, but a lot of people cannot deal with probabilities. So we have to learn to deal with probabilities.
Thank you.
Another good example is financial advice. Whether or not you invest your own money or whether you go to an advisor. Everybody can do financial investment. You can put your money here or there. But a lot of people look for financial advice. Why do the people look for financial advice? Everybody can do this on his own. That's the difference. So you have physicians.
Anyone else? So thank you very much once again. So we'll now move on to the next